Okay, then I would like to welcome all of you to today's colloquium. And we're very happy to have Virginie van Wassenhove as a guest today. I assume that many of you already might know her, but for those of you who don't know her yet, Virginie van Wassenhove received her PhD in neurosciences and cognitive sciences at the University of Maryland, College Park, where she worked under the supervision of Professor David Pepe and Dr. Ken Grant. And already during her graduate training, she focused on the perception and cortical bases of audiovisual speech, and also proposed a predictive coding framework for multisensory processing. In 2005, she then worked with Professor Sri Nagarayan at the University of California, San Francisco. Later on, she moved to the University of California in Los Angeles, focusing on topics like implicit multisensory statistical learning, time perception, gesture communication, and interpersonal interactions. And since 2018, she joined the Cognitive Neuroimaging Unit, directed by Professor Stanislas Dehaney, where they set up the new Neurospin MEG Center. In 2012, she also became an interim group leader of the Cognitive and Brain Dynamics Research Team. In 2013, she also obtained her habilitation and was granted CEA International Director of Research. And currently her research focuses on temporal cognition and multisensory integration. Also, since 2020, she is an academic editor at the Journal of Cognitive Neuroscience and a reviewing editor at eLife. So lots of contributions to the field. And we're very, very excited to learn more about your research today. And yeah, with that, I would like to hand the word to you. Thanks for being here today.
Thank you so much for this nice introduction, Laura. So I'm very happy to be here. And this is a bit unnerving to me because I've been mostly talking about temporal cognition in my last talks over the years. And so that's the first time in a while since I've gathered most of the multisensory work into this talk. So I hope I will not stumble onto some of the new results actually that are included here. Also, as Laura said, please feel free to interrupt me during my talk. I'm fine with this. If you have questions, if you want things to be clarified, I'm perfectly fine with being interrupted. So don't hesitate at all. Okay, with this being said, I always, most of the time, start my talks by thanking the people who actually did the hard work. So at the top row here, you have all the people who've contributed to the work I'm going to show you, and I'll go one by one. And as I was introduced with my PhD thesis work, we'll start with that. So talking about attention in time. So I actually started my PhD about 21 years ago, which was something I had forgotten yesterday when doing my slides. And I did work with David Pepe and Ken Grant over right there. And what they did is throw this McGurk illusion that you probably all know, but just for the fun of it, I'll do this here. And I don't know if it works for you, but this is a case where you have a TA that comes out. So they threw out this illusion at me and they say, okay, you know, what's with this McGurk illusion? What happens to auditory visual integration? How does it work? And so on. And so at the time, there was basically the basic question, which I think is still alive nowadays, which is, when does multisensory integration occur? So is it an early stage problem or is it a late stage problem? And one of the models that existed at the time that was devised by Dominic Massaro turned out to be the first model, Bayesian model of perception. At the time, he called it the fuzzy logical model of perception. And the fuzziness came from the fact that you could be anywhere between 0 and 1. So it was not a categorical perception. And in this model, this old model, there was this really early stage, which was called evaluation. Years later, the models of multisensory integration complexified. So in 2010 now with a review by Talsma et al., in which you have, you go all the way up to semantic analysis and the congruency matching between sound and vision. And still, you still have this early little thing going on between audition and vision in particular. And so I'd like to talk about this as a first entry point of some of the work we did on this problem of early integration and what would that mean. And this is a study that came about contrasting two different psychophysics studies. The first one was by Fujisaki et al. And the second by Van der Berg et al. In the Fujisaki et al., so they asked this question of, do you have an early or late multisensory integration? In the Fujisaki et al., what they use is a visual search paradigm in which they varied the number of targets, and participants had to tell which visual target was in sync with the sound. And they had different levels of complexity in the visual stimuli, but they were still pretty low-level features in the design that they used. The audiovisual target was changing in synchrony at a rate of about 10 Hertz, so quite fast. Every 100 milliseconds, something changed. And with a series of experiments using these different stimuli, they actually showed that there was no evidence, basically, of early integration. And in contrast to this study, there was the one by Van der Berg that used this sort of stimuli, a conjunction search paradigm, which you can see here, with the task of participants being to find a bar that was either horizontal or vertical. So with this, you had the sound changing in sync with the color of the bar, the target bar, and the temporal frequency they used was much slower. It was around 1 Hertz. And with this kind of task, they actually found or claimed to find early integration. What caught our attention with Ankesem, who was a PhD student at the time, was the wide difference between the frequency that was used by these two tasks. So we wanted to use basically, again, the study by Van der Berg, but we made some variations. So I'll show you again here, and you should hear some sound as well. So this is simply to demonstrate that you do not have a pop-out. I mean, clearly, although everyone wished to see a pop-out, it is not a pop-out. It's quite difficult. And when you look at the video, the target, because probably not so many found it, the target is right here. So the key point here is each time this one changes color, you hear a sound, okay? So what we did with Anne was use exactly the same task. We varied, of course, the number of visual distractors to vary the difficulty. And then we tested the different temporal rates from 0.5 Hertz to 10 Hertz parametrically. And we had three conditions. One, the control in which the vision was alone. Another one in which the sound and the visual event were in sync, as I just showed you. And a third one, and that was a change from what was done by Van der Berg et al., in which the sound was tuned or in sync with a distractor and totally unpredictable of the visual change. And this was not in the original design. And then in the task, we also asked participants to report and identify whether the target was horizontal or vertical, so we could have an index of performance. And so what we found is indeed that the reaction times and both the target and identification and reaction times were basically varying as a function of the task requirements or the conditions, with a particular and efficient integration below 2 Hertz. And how we define the efficient integration here is that the reaction time in the control, so in the visual event, was in between the reaction time that we found when we had a congruent audiovisual in sync, basically condition. And participants were the slowest when there was an incongruency, so when the visual distractor and the sound were in sync. So this was interesting. This is true also of any temporal rate, even though there's a general slowing down with increasing temporal rates. And then also very interesting, and what enabled us to say that this was an efficient integration, was the fact that the performance was best when you had a congruency in time between sound and vision below roughly 1.42 hertz. Of course, we did not test continuously, so we can't tell you the exact number. But basically, the performance was best in that regime, and it was worst for the incongruent. So we had like a double dissociation, so to speak, between these two conditions. So what was interesting with this little piece of psychophysics evidence is that it basically is totally in frame or in line with the framework that was proposed at the time by different researchers, notably the one that's proposed by Mary Jones, in which the idea is that your attentional, that you can orient attention in time by virtue of being entrained by sensory events. And in our tasks, the idea was that the sound were entraining at a particular rhythm so that you could detect the visual event better. Now, what's interesting is that, of course, the feature space in sound and vision was totally orthogonal, right? The sound was only demarcating a particular point in time. It had nothing to do with the bar being vertical or horizontal or changing color per se, right? Just the change of color, the change was what mattered here. And the range and the frequency at which we saw that was quite interestingly in 9, with the kind of neurophysiological findings that Lakatos and Charlie Schroeder has put forth at the time, in which we could see around 1 or 2 hertz, this variability in auditory and visual cortices as a function of a monkey's attention. So going from there, basically the idea is that when you are confronted with this sort of rhythmic stimuli, you can develop a strategy that is a oscillatory strategy, as I mentioned, where you tune in to the particular rhythm that's presented to you. When the target is in sync with the visual target and the sound are in sync, then you highly benefit from the sound. When you're in sync with the distractor, you're not benefiting because the sound is tuning you to a distractor. So you have to recalibrate each time. And if you go over a certain frequency regime, you're basically saturating, right? So you can't follow up anymore, and you're not able to develop a strategy that is rhythmic anymore. So what does that buy you also to have this notion of phase alignment between sensory modalities? With Anne, we also pursued this notion of phase recalibration by using the temporal recalibration, which you probably all know about. This is a phenomenon in which you adapt to the asynchrony between the sound and vision for a certain amount of time. This was a study that was demonstrated by Fujizaki et al. And Vroman et al. And since then, there's been a lot of studies showing that. So when you do this adaptation in time, you're going to increase your tolerance to the asynchrony between the sound and vision. And this translates in either a shift of PSS when you do a temporal order judgment, or in the shift in the width of the curve when you do a simultaneity judgment, as we're showing here. And my cartoonish rendering of this is that basically you have an auditory event, a visual event in the physical world, and they get closer in your mind. And our question was, why are they getting closer in our mind following that adaptation? So since we were looking at this phase response, we were interested in testing whether it would be, again, a question of following response. So if we present a stimulus that is rhythmic in time, you're going to develop this strategy, just like in the previous experiment. And the idea was whether this oscillation that you can generate through the presentation of a rhythmic stimulation would actually endogenously shift its phase response so as to realign auditory and visual events. And so we use this sort of strategy. We had a series of temporal recalibration blocks in an MEG experiment here we're talking about now. The sound and the visual events were desynchronized for about, let's say, a minute for a few trials. And what we were interested in is looking at the phase response before the adaptation and after the adaptation, when participants changed their temporal order perception. We presented with these stimuli at a rate of about 1 hertz, which elicited this 1 hertz response in the frequency spectrum in both auditory cortex and visual cortex. And then we basically estimated the phase response of that evoked oscillation and contrasted the two. So I give a complicated story very shortly here and go to the main point, but feel free to, again, ask me details afterwards or whenever. The two main observations that we found in these experiments was first that the evoked response did not show any change, any major change between the pre-adaptation and the post-adaptation period. So the response, the phase-locked or evoked response did not change, but the phase shift in such a way that it was predictive of what participants reported in their PSS. So basically, here what you have is the PSS of participants following the adaptation period as a function of the changes in the phase response recorded in the auditory cortices. We did not find that in visual cortex, so it's an asymmetrical results. And we found that basically here, if you go the more negative phase shift, means that the more forward in time the sound is being processed, meaning you need a negative PSS, that is, you need to present the sound earlier for participants to report the same simultaneity. So it's sort of a compensatory phenomenon. The cartoonish picture, again, for this phase shift is to say that the temporal frame in which the events are being encoded can be endogenously shifted. Now, you can think of it as also as a Bayesian perspective in the sense that the certainty of visual information being presented to you in this experiment was very high, but typically the sound is being presented to you with headphones, especially in MEG, so that it's centered in the head and it's highly uncertain. And so this phase shift here can be also a means for the brain to recalibrate the sound and the visual event in the same space, whether you think of it in time or in terms of spatial location. I think the two interpretations are plausible. So there are lots of parts in these experiments that are questionable that can be redone. And there's actually Harish Gunasekaran, who's a PhD in the team right now, who's looking at this experiment and trying to do it a bit better and have better controlled, as it was a bit unbalanced. So why is it important for us to think about the phase, and in particular for multisensory integration, is also because the phase response in brain networks are telling us of how the information is passing through different areas in the brain. And of course, many different researchers have been looking into this. So starting, for instance, with Varela, all the way up to Zinger and Pascal Fries, and Marcus Siegel has done a lot also to try to provide some evidence for how to quantify network response and oscillatory response in human brain imaging. So that's what I'm illustrating here. So can we think about an incidence of these phase alignments? So if the brain system has the ability to sort of phase out or phase in these different events, is that of any importance for ecological stimuli like speech? And these are unpublished work that we're still trying to digest and work on right now. So what we did in this experiment is we departed from the observation from several studies now that there is a natural asynchronous between sound and vision in natural speech. So the visual speech tends to precede the sound. It's not always true. So I'm listing here different studies that are being a bit more systematic and finicky about the whole story here. But for instance, one of the highly cited papers, Chandra Zakaran, is showing you here the difference between the time to voice, meaning like the time at which you can detect on the viseme a change in the lip gesture, and the actual sound onset, the vowel onset. So what we did in this experiment is we asked whether the brain was sensitive to both physical asynchronies, like we do for beeps and flashes, like I just presented you, and or whether it was also sensitive to the natural statistics of audiovisual speech. And for this, what we did is basically record stimuli of two syllables that were pa and ta, and we matched the physical asynchrony. So you can think of them as stimulus onset asynchrony between visual speech and the sound. And we also matched their asynchrony with respect to their natural asynchronies. So for instance, if I take the pa that was articulated here, and I were to be a physicist and looking at exactly when there was a change in the acoustics and in the visual phase change, there was in fact 800 milliseconds of difference for that natural utterance between the first time I could detect a pixel on the visual video and the actual vowel onset. So that's a huge asynchrony. Okay, and the design was very simple. So the design was a presentation of the syllables to participants, so 6 of them in a row, and then they had to detect a target at the end, but we did not analyze that. So what was interested to us was just the brain response to this series of stimuli and syllables, and this is the range of asynchrony that we actually used. So again, I'll go through the sort of main points and not through the entire details. So some of the early results here are partial replication of what we already know a bit in the field of speech asynchrony. So if we look at the evoked response, the auditory evoked response, we found a decrease of the amplitude of the auditory evoked response as a function of SOA, so physical asynchrony. And we also found a decrease of the auditory evoked response as audiovisual speech gets closer in time, so closer to their natural synchrony. And this is in line with the several studies now in the field of the auditory evoked response. This has an implication also for predictive coding. So these are all of these synchronies for all of them. And then here you see them distinctly for PAR. The A0V is the natural asynchrony for PAR. And you can see that the amplitude is less than the one that is preceding with the visual, and it's still less also when the visual precedes, which is natural for the PAR syllables, and same thing for the TAR here. Okay, this is for the simple auditory evoked response that is locked on the auditory speech. Then what Nikos Halas did is do a very fancy functional network analysis on all of these results. So you see the full spectrum here, but I'm not going to go through all of it. The main gist here is that we do see the implication of many different nodes and frequency, oscillatory frequencies in the analysis of audiovisual speech. And the particular analysis enabled us to look at the nestedness, meaning that the high frequency activities tended to depend on the phase of the low frequency activity. And one of the main results that Nikos found was in particular the engagement of a delta, so the phase response in delta, that was going in a particular direction, namely from more frontal and motor cortices downstream to the SMG and STG auditory cortices, parietal and auditory. So he did a phase information transfer analysis indicating or suggesting that basically the responses in the low-level regions of sensory cortices might be modulated by higher-level regions and by low frequencies. So this is still work that we are analyzing. I don't want to be too conclusive on this yet. Okay, so this notion of phase response is interesting for the communication across oscillatory networks. And deep down, what it really means also is relying on a very basic notion that is essential for multisensory integration, but for any neural integration, which is that of temporal coincidence, right? So the fact that any neuron at a single unit or larger scale may be very sensitive to the fact that things or information happens at the same time. And the ability to vary the phase endogenously enables to modulate this, but we can have also a more hardcore approach to this. So I'm listing here because these are interesting papers and I'm just highlighting them to you. But so the notion of temporal coincidence, as I was mentioning, from neuron to circuit can be thought of and nourishes a lot of thinking in terms of a time domain. It has been historically implicating in audition. And more recently, we can start thinking about it in multisensory regions, because as soon as you start engaging a system into being coincident in time, then you can pass on information, integrate, and so on. And one of such model in multisensory domain is that of Sigari Parisi and Mark Ernst that was proposed a few years back. that essentially relies actually on this notion of temporal coincidence and inspired by retinal cells, but they applied it to this notion of multisensory integration. So here you have an extract from the original model in which you have the visual input and the auditory inputs that are going to go through some form of temporal filtering, so two separate channels. And then eventually you have a cross correlation going on between these two channels that will give you two outputs. One, which is the correlation detector, so how well correlated in time those two signals are, and how delayed they are in time. So the correlation and the lag detector, from which you can derive the impulse response function here that is being provided. So as the lag increases, you're going to have highest correlation. As the lag change, this is somewhat arbitrary. So here is light first and here the sound first. You're going to have differential response between the order of these two responses. So we departed from this observation and we were bold enough, and in particular, Jacques Pénaud-Le-Rousseau, who is now a PhD student in Marseille, was bold enough to want to test the hypothesis of the existence of a correlation detector with MEG. So take a model that exists at the low level system and let's test it on the large scale system. So we, of course, asked some help from Cesare and Mark, since it was their model. So we made sure that we understood what we're doing. So here what you have is the sequences that were selected for MEG testing. So Jacques chose 6 sequences. So in the original test, what they were using, instead of using a single sound and a single visual event, we had a sequence of 5 sounds and visual events. And participants have to tell whether on average in the sequence, in a temporal judgment task, whether the sound is first or the visual is first. And in a causal judgment task, whether the auditory and the visual events come from the same source. Okay, so it's a Bayesian inference kind of thinking here, which is very close to a simultaneity task really, but we respected the original findings. So the sequence was selected to try and maximize the difference between these two tasks and to try and have a good spread basically of the correlation and MCD lag outcomes. So here's what's represented is in fact, the performance of participants in the MEG as a function of the outcome of the MCD core and MCD lag. So MCD core is, again, the correlation. The lag is for what provides you with the order. So participants did this task while being recorded with magnetoencephalography, meaning that we had brain recordings with this. When we look at simply the evoked response, which was basically locked on the onset of a sequence, and simply look at the grand average, so it's a very coarse measure here, we do find a very strong difference between doing a causality judgment and an order judgment in favor of the causality. So the amplitude of the MEG response is much higher when you do this causal inference kind of judgment. And the engagement is highly parietal. So you see, again, here, this is MEG, so we do a source estimation, and that's relatively rough compared to what you would get with fMRI. So it's always with cautionary approach here. But we do see the engagement of a lot of parietal region and some of the well-known multisensory parietal STS, posterior STS, sorry, regions. What this little graph tells you here is that on a single trial basis, just looking at the evoked response, we could get, we could easily tell that participants were doing a causality judgment. So the engagement of the response here were predictive of the task participants were doing, in the sense that we could tell they were doing a causal judgment, but we couldn't find the same results for order. So it's already seemed that it was easier to detect the engagement in one task over the other. Now, the crux of this approach lies in the methods here. So what we did now is use a well-known method, which is the multivariate TRF, or multivariate temporal response function, that was essentially put forward by Cross and Lawler team that has been used in many domains, including speech processing. And that enables to basically try to extract the temporal response function that's responsible for the elicitation of a particular brain response, given that we know about the time course of a stimulus. The benefits that we have with the MCD model, which is that we used here, is that we have time responses. The model provides us with time series. So what we thought with Jacques is that we can not only use the visual sequences and the auditory sequences in this MTRF, we can also include the time series that are produced by the model. In other words, have what we call the time-resolved encoding model. So what we used with this MTRF is not only convolving with the stimulus features, but the predicted outcome of the MCD core and the MCD lag. And that's the key point here, because no other model, if you don't have a biologically plausible model, you can't do that, right? You would only have like one value or a single feature, whereas here we really had a time course. So that was the novelty here that we include in this task. So basically this is saying what I was just summarizing what I was just saying. You basically typically use the stimulus and then you convolve this and then you try to predict whether you have a very good measure of this in the MEG response. So of course we did this. We did this for the auditory sequence separately from the visual sequence. And then in the model, we also used the outcomes of the MCD core and the MCD lag and checked whether we could find an MEG response that was plausible to reflect either of these two outcomes. So the outcomes is here. It's quite straightforward. So here are a bit of our sanity checks. We do find responses that can explain the auditory stimulus. We can find good response in MEG that account for the visual sequences. And when we look at the cause, so the MCD core response and the order, the MCD lag, we have a bit of a more complicated result. So here what is being presented are the Pearson's correlation scores. And you can see that in both tasks, we had a very high significant correlation score for the MCD core outcome, but only in the order task could we find the MCD lag outcome. So there is a bit of an imbalance between these two outcomes. So we discussed the matter in the paper that's been submitted because we tried many different ways to try to see whether this was a real difference or this is a lack of SNR and so on. And of course, more work would be needed. But so far, it looks like the MCD core is more robust in terms of outcome in the MEG. So it does mean that the correlation between sound and vision can be extracted out of the MEG results in conformity with the predictions of a model, but the MCD lag is a little bit more difficult to extract. And the regions that we found that were activated and implicated are again quite far-ranging. They include visual and auditory cortices, as well as a lot of the posterior STS in particular, you can see here. How much time do I have left? I'm extremely bad with time. And
you have like 5 to 10 more minutes.
Okay, so I'll try to go on to a last experiment, maybe not in all of the details, but I'll try this. So one of the importance, I'll go straight here. One of the importance also in speech is that of notion of temporal comodulation. So we just talked about temporal coincidence, and temporal coincidence is convenient when we have very discrete and transient signals. But in lots of these multisensory signals that we're talking about, and in particular speech, we're going to have continuous, much more complex signals. And that's where temporal commodulation comes into play. Here you have some of the earlier work that Ken Grant had done. For instance, where you can see that the area of the mouth movement is highly correlated with particular frequency bands in the acoustics of the speech signals. And we believed already at the time that this probably was an important multisensory cues for the integration system. So instead of using speech, we wanted to use something that was a little bit more tractable and parameterizable, so to speak. And we made this complex study, which I'm going to try and simplify here. And I'm just realizing that the timing has been screwed on that slide. So I apologize for this. Okay, so in this task, so probably most of you are familiar with this notion of random dot kinematograms, in which you have dots on the visual screen that are going to suddenly switch to a particular coherence motion and go in a particular direction. So we departed from that, and what we did here is two populations of dots that have each a different color, red and green. And the task, the basic task, is which of these two population is going to be more coherent than the other one. It's not about the direction. So the direction was always varied. We just wanted the coherence. And the direction could never be left or right or up or down. It was never that easy. It was really random. So I can show you here. Unfortunately, you might have the sound at the same time, but that's okay for now. Sorry. So it should be extremely difficult for you to tell. It's a very difficult task, but you get the gist, so you have these dots. Okay, how do I spell that? Okay, so imagine the same thing without the sound. This is the basic task. Then we add the sound. So what you just heard are actual sounds, or what we call acoustic textures, that are exactly matched to the dynamic of the most coherent visual cloud. And so each visual cloud would start with being totally random. And then at some point, 500 milliseconds later, they would start taking a direction with a particular degree of directions. So it's typical in motion coherence from 15 to 95%. And so each dot was matched to a particular frequency sweep. There was no change in amplitude modulation. So it's not a full acoustic textures. But so if a dot started at the bottom of the screen and going up, then the sound would go the lowest frequency possible in the spectrum all the way up to the highest frequency. Okay, so the matching was done in that way. Now, as you could tell from the video I just showed you, it is not a pop-out, right? It's even very distracting. You probably weren't thinking that you would have sound like this. Okay. Now, what did we do with this general experiment? So of course we could test because we had different degree of coherence. We could build a psychometric curve onto how good participants were at detecting visual coherence or discriminating visual coherence in that case. We had three groups, so it's an MEG experiment. We had three groups, one who did this solely visually, one who did this with this correlated audiovisual that I just explained, and one that had auditory noise, distracting noise. And the key point here is that all of the participants, whatever groups they were in, had a pre-training session in which they all did this with visual stimuli only. Then they got a training, and then they got a post-training again, and the post-training is again visual only. So all of what I'm going to show you is always with participants who are looking at visual stimuli only, just their history differs. Some of them will have had training with sound and vision, congruent sound and vision, and some of them with noise, okay? So first the performance. So of course our hope here was that the audiovisual congruent training would benefit the performance on this task, which is effectively what we showed. So here you have a psychometric curve before in light gray and after in the post-training. For the visual group, the congruent audiovisual group and the control audiovisual group, you can see that all three groups perform better after being exposed to this task. But most importantly, the perceptual threshold for the audiovisual comodulated group was much more improved than for the visual group, which was nearly equivalent to the audiovisual control group. All of them had improved reaction times, all three groups, with comparable improvement in reaction times. And even though the audiovisual group was the one that improved the most, they were not aware of having improved. So only the visual group explicitly improved their confidence rating, which is found interesting. Okay, we were interested, of course, in the MT. So for sake of time, I'll go fairly fast here. We had a orthogonal source localization with MEG, so we could localize on each individual the actual regions that were specialized to the coherence level. And here you have basically the neurometric. So we looked, I'm going to show you right away this full slide. So here you have the amplitude response in the HMT plus region as a function of the coherence level. So the bluish is the lowest coherence, the reddish is the highest coherence before training and after training. And for each group on the rows. So for the visual, audiovisual, and control group, you can see that before training, as expected, the higher the coherence, the more amplitude, but it's not really easy to discriminate. And then in the post group, you can see that it's much more graduated, gradual in terms of amplitude response for the visual group. It is also pretty good for the control group, but somehow, and surprisingly, it looked a little bit less discriminable for the audiovisual group that was of interest to us. And when we built a neurometric, which was essentially taking the mean amplitude over a certain window of, over a certain latency as a function of the coherence level, as you would do in behavior, what we found is that there seem to be two computational strategies, so to speak. I mean, this would, of course, need to be tested in probably with monkey neurophysiology, but for the visual group and the control group, there seemed to be maximization of a difference at the extreme of these curves, whereas for the audiovisual group that was of interest to us, we actually found a significant shift in the threshold of the neurometric response. And so this is, of course, having a zoom on a particular region, but since we're doing full brain neuroimaging, then we want to know what's going on everywhere else. So this is the beast with MEG that we can have. This is the visual group and audiovisual group contrasting the pre and the post. And you can see at roughly the same latencies that you have widely different kind of responses here to the same, again, to the same visual stimuli, right? Because both groups here are contrasted with only visual stimuli. So any difference can only be accounted by the history. And one of the observations that caught our attention, beside the typical culprit of multisensory regions, was the regions in the PFC, ventrolateral PFC, that you see here, that showed some significant differences across all three groups for those particular coherence level that were learned by participants. So those coherence levels that were above threshold before training and that passed on being below threshold after training. Across all three groups, this specific coherence level were eliciting higher activity in VLPFC. And that's where we started thinking that perhaps this notion of reverse hierarchies were interesting here. So basically, we're asking the system to extract statistics between sound and vision that are quite abstract, right? Coherence level across a space that is randomized in direction and so on, that participants have to categorize. And somehow these seem to implicate the prefrontal regions that may actually modulate in a top-down fashion the forward volley of information. So this is something that we could test in part, and I'll conclude on a couple slides of functional connectivity. I had included this slide here by saying that, of course, nowadays we talk about Bayesian inference that are going to be much more hierarchical and high level than what we started with Dominique Massaro's. And within these Bayesian frameworks, you need to have some top-down and some priors. And the idea is that this sort of abstract representation that can be afforded by multisensory coherence could feed these sort of priors in terms of amodality or supramodality. So when we apply on those same data functional connectivity, that Daria Larroca was the first author on this work, and these are the last slides of data I will show you. What we did see, so we saw some very nice time frequency response locally that were predictable on the basis of what we know in monkey neurophysiology. So for instance, here you have a depiction of the very, very strong decrease in alpha beta that could be seen in the occipital regions, along with, at the same time, a very high response. So this is MEG, so we can see high gamma response above 60 Hertz quite clearly. What was interesting here is that irrespective of the group of participants, we did not find a correlation between alpha activity and the motion coherence, but we did find a strong correlation between the high gamma activity, so the feed-forward volley of activity, and the motion coherence, and even more strongly after training, because after training from the gamma activity, we could predict the performance of participants, and this is the correlation that you have depicted here. So these are local changes in time frequency responses. When we look now at connectivity across the three groups, so again, the three groups are in the rows, and here you have the frequencies. What's striking here is that you see an increase of alpha connectivity, functional connectivity, in nearly all three groups, which indicates that attention cannot account for the kind of behavior we see here if we take for granted that alpha is an attentional regulation of information flow. The key finding in this functional connectivity was rather this beta. So what was unique marker for the audiovisual group was a very strong beta network that emerged in the post-training as compared to all the other three groups, as you can see here. And in particular, the beta activity that we found in this functional connectivity was correlated with the confidence that participants reported in their post-training behavior. Sorry. So beta activity is important because in different lines of work, it's being shown that it may actually convey top-down information, but in a content way. So the recent work by the lab of Earl Miller, for instance, would suggest that prefrontal regions are going to be providing some content-wise or working memory-wise information to downstream activity. So the idea here is that you may have information that this notion of temporal comodulation or temporal coincidence across sensory modalities that can be a very strong driver for multisensory integration in a bottom-up fashion, but it is not enough perhaps to just appeal to attention to be a driver for strengthening this comodulation. And we may need to appeal to more abstract representation that could be afforded by areas like PFC in such a training task. So my take-home message are those. One of them that I just mentioned, selective attention, which I would contrast with top-down abstraction. Of course, I come from the multisensory side, I come from more the speech domain, so we're not too afraid of talking about abstraction, but I agree that it is much more complicated once we go into perception if we try to avoid notions such as conceptual knowledge, for instance. And the idea, of course, is that being able to address this notion of abstraction can give way to, or give access to thinking about Bayesian priors as a model representation that can come and constrive the kind of multisensory experiences we can have. And on this, I will thank again the different people I mentioned, and thank you for your patience, as I think I've run over time.
Thanks a lot for this great talk. Very, very interesting. We have lots of virtual applause. Okay, those of you who've attended our colloquium before know that I like to sort of structure the question and answer session by asking you to put your name in the chat if you have a question, and then I can just call you to ask your question, or if you prefer to write your question in the chat, you can also do that, and then I will read it out loud. So maybe while people start thinking about whether they have questions, I will start out with a question. In your, when you were talking about the multisensory correlation detector, you mentioned that one of the big advantages is that both the model as well as your MEG response is, of course, time resolved, right? Mm-hmm. So when you find that, in particular, the core component of the model predicts variance in the MEG signal, does your analysis also tell you sort of where in time it corresponds the most? So would that speak in favor of like an early or a late integration process?
Yes and no. So I vaguely remember now. I don't have slides or figures to show on this. We had a lot of discussion with Jacques, who essentially is the working force behind this, about trying to pin down the latencies. The complexity here is that the sequence itself was already quite variable. So we have very strong transient responses that may get in the way of much more subtle responses. So we have a trade-off between being able to actually test the model and hence adding some variance with respect to the stimuli and having this discrimination threshold, and the actual latency at which we would have a maximal response for each of those components. So yeah, I think ideally that's the way we would dream of going towards, because we would love to have these components. And there's probably ways of tricking that and of tweaking the experiment in such a way that we can get that refined. Since it was a first for us to try and be that wild, because it was really something we didn't believe we'd be able to get results of, I'm quite happy, but I think there's much that could be done. I mean, it can open also the way to source localization driven, right? So we could think of mixed populations in which we try to tease out the particular tuning components and try to restrain the source localization on this. Yeah, so the short answer is it would be wonderful, but right now we're a bit limited for that particular study. So it would require more investigation. Yeah.
Okay, cool, thank you. Maybe another question that comes to my mind, and I don't mean that to be a mean question, because I know that the focus is on time here, but of course, to me, as someone coming from more of a selective spatial attention background, I of course wonder, especially when the task is to determine whether a sound and a visual stimulus comes from the same source, then it would be intuitive to me to think that, of course, spatial location should also be like a critical factor.
Yeah.
So, yeah, do you have thoughts on that in general? How maybe like the temporal and the spatial aspect go together here? I don't remember, but I think in your task, it was just one location, probably, right?
Yeah, yeah. I think both of them are of course essential. I think space has been more early on in multisensory research. The spatial dimension was the one that was most studied and explored. But strangely enough, I think there are major differences between animal work and human work. So once you get contrived to an fMRI or to an MEG system, you're not going to, very few labs are capable of adding some spatial dimension in terms of real dynamic changes for the sound in particular. It's easier for vision, but to try and accommodate for really good auditory specialization, it becomes complicated. And also for tactile, right? Because you're going to not want to move and so on. This being said, I think that looking just at the angle of spatial attention will bring you to the problem of where things are. But once you know where things are, and within a certain range, because of course there's variance there too, then you want to assign meaning to what it is that's in that region. And so sometimes I take the toy example of if you happen to have a person with a dog and the dog is barking and the person is talking, you don't misassign these things, even though the signals from a pure bottom-up approach, they can be very well mixed. right? They can be in the same spatial region and so on. So there has to be a little bit higher selectivity. And the argument here is that this selectivity may not just be about attention, but also about meaning at some level, and meaning assignment to the kind of signals that are constitutive of what your brain is analyzing. The coherent dot motion task includes a little bit of this notion of spatial selectivity in also in a bit of an abstract way, right? Because we have this coherence and we don't really know phenomenologically when you do this task, you sort of have you sort of feel that there's one direction that's more coherent or whatever, but it's not really clear. So you also have this level of that's a bit fuzzy in terms of spatial assignment. That could be interesting. And I don't know whether I would go as far as saying that this is attention. I don't know about that. No.
Okay, thank you. Okay, I'm just going to keep asking questions because no one writes anything in the chat. Just a quick follow-up on the first study that you showed with where they had to judge whether the bar was horizontal or vertical, and with the beeps that were either synchronous or asynchronous. Did the subjects know that the beep, were they instructed anyhow on like the relevance of the beep, or no, is that?
Yeah, they were instructed that you will hear some beeps, but you don't really care. So they were told to neglect basically the beeps. But at the same time, and I went very fast on this, at the same time when the display occurred, the sound started before the display. So we basically we entrain participants without them knowing it, right? With 2 or 3 beeps before the actual sound visual stimuli arrived. So I think these sort of studies for me remain always, including the one that Van der Berg initiated, remain quite ambiguous in their interpretation, because you clearly pay attention. I mean, you clearly direct a particular fluctuation in the system if I look at the brain response itself. And so you orient a particular encoding windows to be at the right time, at the right moment, so to speak. And should we call this attention, or do we call what, if it's not attention, then what do we call it? And then if that's an attentional problem, then in the early late dichotomy, it doesn't make sense anymore, right? We need to call it differently, because if attention is there, then it becomes a late process in a sense. So the terminology, I think, needs to be fine-tuned on that. It's a bit complicated on these models.
Yeah, that's right. Okay, we now have a question by Louis.
Oh.
Hi. So I really enjoyed the talk. Thank you very much. Just took me some time to get my thoughts in order. So it sort of clicked for me when you just said, but is it attention? And I thought that perhaps I wondered the extent to which your experiments on causality or causal inference and temporal order judgment could shed some light into this single term that seems to conflate everything, right? So to what extent would you say your results actually give some insight into perhaps separable aspects of attention with regards to, in this case, cueing, right? Or to, as what Mark would like to say, attention is just gain, right?
Yeah.
So do you think, so what insights do you think these experiments could shed in terms of distinguishing between these different terms for attention, or how attention might be implemented?
Oh, that's a hard question, actually. So I would say that what strikes me as very interesting is if we push the envelope and say, okay, you have coincidence detection, and this is what drives integration at different stages, then we end up with this notion that all the information that correlates in time and space is going to be integrated. So obviously you have a need for a selective mechanism that's going to act as a decorrelator, so to speak, right? Everything cannot be correlated without being at some point decorrelated. So I think it's fundamental. I think Horace Barlow was using already this distinction early on for low-level vision, this problem of correlation, decorrelation, and the balance between the two. What surprised me in the results, and again, this is the first study looking at it in this way, so I can't go too far in terms of interpretation, but in terms of empirical results, the fact that the correlation was there systematically and quite strongly, but that the order, the lag, was not, seemed puzzling to me, because it's almost like you had this bottom-up correlation going on quite strongly, a very strong drive, and then you really need effortful things to be able to decorrelate. So it still is in tune with this notion of attention, right, of top-down. Whether it is attention per se, I don't know. And what define, and this is more like the speech part for me, what defines the window of time within which you consider things to be correlated or not is another question, right? And in the multisensory, from the multisensory pure temporal tuning of neurons, there are not that much work actually that looked at the simultaneity window. But from what is known, you have a wide array of acceptability. So you can have multisensory neurons that are going to be not so happy, if things are above 100 milliseconds, but some of them can accept as much as 3 seconds, let's say. And it's even wider when you go in prefrontal cortex. So to me, that remains puzzling, because it seemed to me that attention is just not enough. And we need to appeal to especially in humans, to appeal to some form of abstract representation. And I don't know what the nature of that representation is. So that's for me, that's a question that so now I effectively turn to time and temporal cognition much more in the work of my lab, but I still have this problem of how to reconcile this bottom-up, top-down, but from the more abstract meaning. And multisensory always seemed to me the most interesting way to try and tackle that question, because what decides in the brain what should be integrated or not? And time-space are great. There are big dichotomies to be made, but at some point it has to be more refined. And I don't know how, aside speech and linguistic systems, I don't know how far conceptual knowledge can enable to answer this, right? Even if that's enough. So I think more models, more theoretical approach is needed there beyond attention.
So perhaps a lower level and more pragmatic question with regards to how you design the stimuli for these 5 trained visual probes and 5 trained auditory probes. Yeah. So your participants were always aware that you would have 5 stimuli of vision and 5 stimuli of auditory, and they would be jittered, but they would always be surrounding one another.
Yes.
Right? Yeah.
Yes.
So how do you think it would actually be? So what's actually the parameter that you're actually manipulating when designing these stimuli? The jitter between them? Or?
Yeah, so what Jacques did is essentially, because we had an array of sequences that can be used. And what he simply did is in a pilot experiment, sorry, just a second. In a pilot experiment, he tried to separate as best as possible the sequences so we could orthogonalize the causal and the ordinal judgment, and the order judgment. As you could see on the graph, it was a bit more complicated also for the order, right? The sequences were more grouped at the threshold. I can try to, I don't know, if I, how do I do that? Let me see. Can you still see my slides if I do that? Yeah. So here you can see that nearly for the causal judgment, the curve is nearly perfect with respect to the predictions of the model. I'm not seeing the person who asked me the question anymore. Sorry, it's a trade-off between what I can show you. Here we go, Lewis. And so goodness, on this, you can see that the sequences are really nice spread and can allow us basically to have a good estimate of performance at different degrees. But it was much more difficult, and we were much more towards the threshold when it came to the temporal order judgment. Although these graphs here are telling you the performance as a function of the MCD, so as a function of the model outcomes, right? And not as a function of the actual stimuli here. And I guess your question is perhaps that you would like to see this with the stimuli, and I don't think I included that in my slides, but I think in the paper, so this paper is on bioRxiv. It's been pre-submitted, so maybe it's in the paper. I think we had a figure with this and with the individual psychometric curve as well. So you can see the spread.
Mm-hmm. Thank you very much.
Yeah, thank you for your question.
Okay. Since I don't think there are any more questions from the audience, I think we can come to a close here. And I would like to thank you again very much for your interesting talk. And thanks for being our guest today. Just as a final thing, if there are IGSN students in the audience who would like to get a certificate of attendance, please stay with me till the very end so that I can note down your names. And to the rest of you, I hope you have a fabulous evening and a good rest of this week. And I hope to see you again sometime soon for one of our next upcoming talks. Thank you all. Bye-bye.
Thank you all. Bye-bye.